Yes. Thanks everybody for coming. Thanks so much for inviting us. It's a pleasure to be here. Toronto's a great city and I hope to come back more often. So I'm going to talk about bDNA. The commercial name for the technology has been called QuantiGene pretty much since the origin. And it has a number of different applications, mainly doing with gene expression analysis that we'll look at today. So there are three broad families of technologies that use bDNA, branched DNA. You don't know what it is yet, but you'll learn all about it, more than you probably want to know in a few minutes. QuantiGene is a gene expression family for either single or multiplex analysis of both RNA as well as DNA. PrimeFlow is used to look at RNA together with proteins in flow cytometry. And then related to that is actually ViewRNA, all three of which use this branched DNA signal amplifier. And in the case of ViewRNA, it's to look at either cells or tissues localization of RNA. Oops, I forgot I had this. So the original QuantiGene technology is a single-plex assay, very much like an ELISA, which gave you the ability to look at one target per well. I came on board in 2003 and started working with the single-plex assay. The small company that I was with, you may not have heard of it, it was called Panomics way back when, developed a multiplex assay on the Luminex platform. And as time went on, Panomics was acquired by Affymetrix. You know them as a microarray company. Affymetrix was acquired by Thermo Fisher. So QuantiGene is really just a tiny part of what Thermo Fisher deals with. As you know, it's a huge company. But they love the technology, and that's really why I'm still here today. So bDNA, QuantiGene, what makes it unique? In terms of gene expression analysis, the strategy is to amplify the signal, not use PCR or anything else to actually amplify the target itself. It's a combination of signal amplification and specific hybridization that allows you to do gene expression analysis. It actually has been around for quite a long time, since the 90s. It was developed at the San Francisco Bay Area company Chiron. They were looking for an assay to use in conjunction with compound screening, where they could basically take cell lysates, lyse and go directly in an ELISA-based, similar to an ELISA, but look at a target, mRNA target, together with compound screening. So they developed a buffer that was compatible with subsequent hybridization events to allow detection. That technology moved forward and actually became the basis of not only a screening product for single-plex assay, but also as a clinical product for HIV and HCV assays, initially by Bayer Diagnostics, and then Siemens has acquired them. So it's still being sold today as a clinical product, but we're looking at it as a research product. So you're going to see a lot of squiggly lines and dots today. This is what we would call a tree. And the tree is made up primarily of very long single-strand DNA oligos, some of which constitute a probe set. Oops. A number of probes with a number of short DNA oligos, single-strand oligos with a number of functions that we'll go over. There's a preamplifier, which is essentially the trunk of a tree, the branches of a tree, the amplifier, and then there are binding areas on these branches for label probe. And this label probe can be an enzyme, it can be a fluorescent marker. This tree that we're forming is actually going to have 400 binding sites for label probes. So that's our source of signal amplification. So we need to somehow get the signal amplification associated with the target that we're looking to measure. So again, the whole concept of the QuantiGene assay, bDNA assay, in its original iteration was lyse and go. No need to purify RNA, no cDNA step, no PCR. Work directly from cell lysates, tissue homogenates, difficult sample packs such as FFPE and whole blood. Of course, it works with purified RNA. And again, so the strategy is signal amplification versus actual target amplification. And we feel that that helps avoid certain biases, especially with formalin-fixed material. The workflow is very simple. Again, very much like in ELISA. The original formats were in 96 well. We've since introduced 384 well. It still serves as a clinical assay for HIV, HCV. It's been demonstrated to give results that are equivalent to real-time PCR and microarrays. And one thing that we'll look at a little bit more is the linear versus log-log analysis in terms of our ability to measure very small changes. We have formats that will go from 1 to 80 targets per well, and then we have assays, again, for in situ hybridization. So we're going to start with the original single-plex assay. This is actually second generation. We won't bother talking about first generation. There's a lot of, again, dots and squiggly lines here. So we're starting with a specially coded 96 well or 384 well plate, every well of which is going to have the same single-strand DNA capture probe. We have a probe set. We have an RNA sample along the top. And then we have amplification reagents. So of everything that you're looking at here, everything is generic except for the probe set, which is used to capture via hybridization a single RNA target. And this is how it works. So we've got our coded plate to which we add our sample. Again, this can be a crude lysate. And we add a probe set. So probe set is a family of oligos that have basically 3 different functions. There are areas of unique sequence that you see here. These are called capture extender probes that are binding to the target. And then each one has the same universal tail. These are all single-strand DNA. It's going to hybridize overnight at 55 degrees. We have an overnight hybridization for capture. At the same time we're capturing specifically, we're planting down what I'll call the roots of a tree. These are label extenders, which will be the source of binding for the signal amplification tree that we will build. The areas that these are binding to are found only in this target. Our bioinformatics people do the probe design. All we need from you is the target you're looking for. What you get from us is a vial of probe. So we have an overnight incubation, and these, I didn't talk about the small green sequences there. They're called blocking probes. They, as compared to the other two, they're not necessarily target specific, but they're there to help stabilize the structure. So imagine single-strand DNA is going to be pretty fragile. We have DNA coupled to it. It's going to increase the stability and perhaps melt out secondary structure. So we have an overnight incubation at 55 degrees. Via hybridization, we capture specifically the one species that we want to quantitate. And then we start building our tree in steps. So we have the preamplifier. So whereas the bottom parts of these probes are unique to the target, the top of them together as a pair provide the binding sequence for the trunk of the tree, the preamplifier. That's added in excess. What binds, binds. What doesn't gets washed away after an hour incubation. We have a second 1 hour incubation to add the branches to the tree. This is the amplifier. And then along those branches, we add label probe. In this assay, label probe is an oligo, again with complementary sequences on those branches, attached to an alkaline phosphatase. That alkaline phosphatase, after the addition of chemiluminescent substrate, is used to measure luminescence. So we need a microplate reader capable of reading luminescence. The intensity of the luminescence is directly proportional to the number of molecules that we've captured until we've reached saturation. So again, we're looking at a single target. And one of the reasons why we're using what we call the double Z approach for signal amplification is that it adds a great deal of specificity to the assay. The original QuantiGene assay did not have this. This was something that we added during product development for both this technology as well as for the in situ hybridization technologies. But without going through everything in detail, it's absolutely required that both of the pair are present in order to generate a signal. If we have no probe, if we have a probe to a target that's not there, if we have only left or right members of that Z, we get no signal. The actual probe design is interesting. So the cartoon that I showed you was kind of an underestimation of what's going on. Our probe sets for this assay are typically covering about 500 bases. And this is the entire sequence for human TXN2. And in color, we see where our probes are actually binding. So in red for captured, in blue for signal amplification, and then in green for the blocking probes. You don't need to worry about that. We just need to get the sequence from you, or the target ID, and we generate the probe set. And I keep forgetting I'm holding this in my hand, and I'm going over to this. Okay, I'll try and remember. So on average, there's 6 each of the captured probes and the labeling probes, variable number of the blocking probes. An offshoot of this technology is the fact that it's extremely accurate in measuring small differences. So some people might not care. They just want to know if something's gone up 200%. Other people, and this product really came on board together when RNAi was really making a big splash, and people were trying to accurately measure knockdowns or the effect of the therapeutic on knocking down a target. So this is an example of looking at first untreated and then PMA-treated HeLa cells. And what we're looking at is IL-8 induction from PMA. So we're not looking at raw data, we're actually looking at signal relative to housekeeper. So in this assay, in order to normalize, you need to take your sample and split it into 2 wells, one with probe set for target, second with probe set for the housekeeper to allow you to normalize for sample input. So we're seeing the ratio of IL-8 to cyclophilin B. We induce the cyclophilin B, and then we transfect with an siRNA directed to knock it down. And we measure gene expression at 4 and 8 hours post-transfection. We can see that after 4 hours, we have an 87% knockdown, and after 8 hours, we have a 94% knockdown. So 7% difference in expression based on the accuracy of the assay was truly statistically relevant. So that small of a change can be measured with the technology. Of course, TaqMan for single-plex assay is much more commonly used, real-time PCR. Here at Thermo Fisher, we love the TaqMan assay. And even well before my company had anything to do with Thermo Fisher, we showed concurrence of the QuantiGene single-plex assay with the TaqMan assay based on an FDA-sponsored study to look at concurrence between different gene expression technologies. We're going to move to the multiplex assay, which I think is even more powerful. It uses the Luminex platform. So it's very similar in concept to the singleplex assay, but the Luminex assay affords you to use multiplexing. And the way that they do that, now I apologize if you know this already, but the Luminex assay, also called XMAP technology, has to do with the fact that their original assay had essentially 100 different color beads. Each of these beads has a ratio of 2 dyes in it that are slightly different, giving each one a spectral zip code or barcode that is unique. Beads, so they call it XMAP. X is the biomarker that you're looking for, and MAP is multi-analyte profiling. The beads themselves are coded by carboxyl groups that can be used to attach antibodies, or in this case, oligonucleotides. And let's say we're coding our bead with an antibody. We use that to capture an analyte from a complex mixture. And then we have, say, a secondary antibody that has a fluorescent marker that the instrument can also use for quantitation. The Luminex instrument actually, there are 3 different types now. The one in the middle is their oldest one. It's been around forever, very common in labs. The Model 200. The FlexMap 3D further over is a little more expensive. It has 3 lasers instead of 2, can do a higher plex. The original instrument could do 100 plex, the FlexMap 3D 400 to 500 plex. And then they have a less expensive instrument that's CCD camera based that can do up to 50 plex. So again, we're going to use one of these 100 beads to capture a specific target, and then we'll have a way of detecting it. And we'll read our samples by putting a probe into the well that contains this reaction. The beads will go rapidly past two lasers. There's a red laser that will ID the spectral zip code of the bead, and we've said, well, this bead number 3 is capturing IL-6 or whatever it happens to be capturing. And then a detection antibody in this case that's fluorescently labeled with phycoerythrin, and the intensity of that fluorescence is determined by the green laser. So the way the assay works is you're not looking at a single bead in the well, you're actually looking at 50 or 100 beads per target per well. And the raw data that you get out is the median fluorescence intensity. So it's highly accurate in terms of giving you median fluorescence intensity. When we're looking at QuantiGene plex, we're switching from antibodies to oligos. And let's take a quick look at how the multiplex assay works. This is actually one of my favorite assays because of the amount of data that it can generate with very little effort. So I've been talking about trees, but now we have Luminex beads. And so we're not using a coated plate, we're using coated beads. And each one of the different beads will have a different single-strand DNA oligo that'll work in conjunction with a probe set to capture specifically single mRNA targets. As you can see here, again, this is an underestimation of what's going on. A bead's about 8 microns, so it's about as big as a cell. This is supposed to represent an RNA molecule. And again, we're typically covering about 500 bases. And the specificity of capture as dictated by the tails of each of these capture probes. The singleplex assay.
Fixed number of probes per target? Or?
It's kind of target dependent.
Because if you're normalizing or if you're comparing across different targets.
Right. So
more probes, more signals.
Typically, the way that the assay is used is we're looking at relative gene expression comparing one sample to another, not looking at actual copy number. We could look at actual copy number, and people have been doing that with the singleplex assay, but it requires running standard curves of in vitro transcripts. So for the most part, people are just looking at changes relative to housekeepers.
And how do you deal with secondary structure? Because RNA is complex.
So this incubation is happening at 54 degrees, so that helps a bit. The buffers that are in it help with the secondary structure, and the blocking probes that we've added help with the secondary structure. The probes are added in excess, and simply binding those sends them towards, being more linear than binding to themselves. But one point that this brings up is the fact that we no longer have to split our sample between 2 wells. We've got possibility of going up to 80 targets in that well. So we can have 1 housekeeper, we can have 5 housekeepers, we can use them all for normalization. And we can look at that many targets as well. So, and I was going to mention the difference here between this assay and the singleplex assay. The singleplex assay, again, based on hybridization, you just need an oven, a static oven. Here we have a heating shaking oven, a small heating shaking oven to keep the beads in suspension. So we got up to here with an overnight incubation, and then we start washing the beads. I'll talk about how that's done in a moment. And we have basically the same type of amplification reagents with the preamplifier, the amplifier. Here's where the assay diverts a little bit. Label probe in this assay is an oligo complementary to the branches with a biotin on it. And that biotin provides a binding site for streptavidin and phycoerythrin. The Luminex green laser is always looking at fluorescence from phycoerythrin. So that's how we incorporate it into the complex. And subsequently read on the instrument. So what makes this assay so great? Same could be said about the singleplex assay. It's lyse and go. You can, if we had cells that were living now, we could lyse them in a half an hour. They'd be ready to go into the assay, and we have data by this time tomorrow. We don't have to worry about purification of RNA. We don't have to worry about the cDNA reaction. and we can multiplex again up to 80 genes. So we can deal with difficult sample types. And it has simple ELISA-like workflow. The probe sets, new probe sets are something that we generate pretty much on a daily basis. Right now we have quite a complete library, especially for human, rat, and mouse. But if there's targets that you need that we haven't made yet, to get them incorporated into a new QuantiGene Plex panel is maybe 2 weeks away.
So, Marcel, sorry to interrupt.
Of course.
So multiplexing is a huge benefit of this assay, which I can see compared to the qPCR. What the sensitivity? Like, did you measure the sensitivity?
I'll show you that in a moment.
Yeah, that will be a question.
Yep. I actually have a data slide that compares sensitivity of the singleplex and the multiplex assay, and then you can compare that to what you believe for qPCR. This is just showing, again, probe design. It's very much similar for the multiplex assay, and nothing you really need to worry about. You give us a list of your targets, and you get a vial with all the probes mixed together, and a second vial with all the beads mixed together. So when you order this product, all the reagents are generic again, except for the probe set and the matching beads. When Luminex technology was first developed, bead-based technology, they did not have magnetic beads available. And washing required using a millipore filter plate and a vacuum manifold. There were sometimes issues with that protocol because a well could potentially not filter well. What do you do with that? You could filter too strongly and the beads could get embedded into the filter mat in the bottom of the filter plate. When they started with the magnetic beads, we were happy because we came out with a small handheld magnetic bead washer. You're using a flat bottom plate. The plate goes on, gets held by the clamps. You wait a minute for the beads to come down, and then you just flick it like an ELISA plate, and all the beads are retained. So it just it took a variable out of the assay and made it more user-friendly.
We do have automated washers too.
Of course.
Which is helps give them more fun.
Of course. So when you want to increase your throughput, when you're running 10 plates of this a day, you want to use it.
Yeah, automated, yeah.
So of course, when we were developing this multiplex assay, we wanted to compare it to data from the singleplex assay. So the simple experiment where we're taking U937 cells plus a minus stimulant, and then looking at inflammation response with a single housekeeper gap pH. So the data concurs in terms of response to stimulation, but probably the most significant fact is that with the singleplex assay, we needed to run 20 wells. For the multiplex assay, we needed to run 2 wells. And we had an internal normalization to housekeeper. This is one of the few real data sets that I'm going to show you again to highlight the specificity of the assay. So we were working with Big Pharma. They were doing a human cells into mouse, human tumor cells into mice xenograft model. And they wanted to see how well QuantiGene Plex assay worked in differentiating mouse from human targets. The same gene, mRNA, obviously a little bit different, but could we differentiate the species in a mixed population? So along the top, you see in red human probe sets, in mouse probe sets in red, human probe sets in green. The names of the targets are sanitized out. But the first row of data, the low numbers, that's typical backgrounds. Average backgrounds are low for the technology. And then signal goes up from there. So if we look at a dilution series of human liver RNA, we see signal for human targets as long as they're present in liver. If we then look at crude homogeneous, naive spleen, naive liver, again, both for mouse targets light up, and human are all at background. And then when we look at xenograft spleen, xenograft liver, everything lights up. So this was a good demonstration of the specificity of the assay, and they were obviously pleased, and we're pleased to show the data. Amgen, when the technology was first introduced, did a comparison with qPCR and showed that this is in LPS-challenged mice spleen using purified RNA, comparing qPCR and QuantiGene data. The data is essentially the same, but the take-home point is that they could go from 14 qPCR plates to 1 QGP QuantiGene plex. Specifications for the assay in terms of sensitivity. The single-plex assay, if you're using a microplate luminometer that meets our specifications, we can reliably see down to 200 molecules with the typical probe design. The multiplex assay is somewhat less sensitive. How that compares to quantitative PCR, I would need to defer to my experts, but what it says to me is if I can see 200 or even 1,000 target molecules and I'm expressing at one target per cell, I should still be able to see it, considering the sample input that I'm putting in. So people have, if I'm using cell lysates, how much do I need? So worst case scenario, if you're using small cells like PBMCs, the maximum input of the lysate is going to be the equivalent of 80,000 cells. If you're using more typical larger cells, cell lines, you might be using 30,000 or less per well. And again, directly lysed and then into the assay. These lysates, the tissue homogenous that you make in a different buffer, can be made and they can be frozen. They can be frozen and thawed multiple times. So you can collect samples, freeze them, and then run them when you're ready. We looked into challenging samples that I hope are clinically relevant. So one was looking at whole blood. And we developed a protocol for lysing the whole blood, similar to making a cell lysate, and then using that directly in the assay. It's great when you use these whole blood lysates. They're bright red, so you always know which wells you put sample into. That's aside. So we could go from a whole blood lysate, QuantiGene, if you're familiar with it, it's another method of stabilizing RNA in whole blood, works as well. If we're using PCR-based assay, we may need to do a globin depletion step, and of course, we typically would do RNA purification, cDNA, and then PCR. So they both work. They both potentially give you the same answer, but we feel that we've shown both here and here that the QuantiGene assay works very well. So what we're looking at here is whether or not the whole blood lysate, a very complex matrix, is going to interfere with our ability to detect the target or not. So this is a dilution series of an in vitro transcript to DAP-B, which is an E. coli target, not present in human. And we're looking at in the background of a whole blood lysate versus just buffer. And in the dilution series, the two curves overlay each other completely, showing that there's no matrix interference. Similarly here, we're looking at GAP-DH in QuantiGene stabilized whole blood. We see nice linearity. And then we go to some papers. So in this case, we're looking at a whole mouse blood, an animal study where mice are treated with an antibody that stimulates the expression of FOXP3. And they're comparing that to non-stimulatory antibodies as controls. Oh, sorry. So the first three animals are comparing both before stimulation and after, and we're looking at FOXP3, and it hasn't changed very much. We look at the 5 mice that were given the stimulatory antibody, FOX compared to pre-bleed, FOXP3 expression goes up quite a bit. One mouse did not show much of a reaction. But again, this is taken directly from bleeds, from whole mouse blood. And it's also been used to look at ex vivo stimulated blood. So whole blood in culture looking at dose response of CXCL10 or heat inactivated. And this was analyzed by the QuantiGene Plex assay. We can see dose response depending on the dose of the CXCL10, variety of different targets seen here. We don't have to go over every little bit of data, but just to show you that the assay works well with blood, either in vivo or ex vivo. And these changes were all the same as that were seen in microarray. When we go to archive tissue that have been fixed in formalin and are embedded in paraffin, there are issues getting good gene analysis. So that was a project that we started with early on, back before Thermo Fisher, before Affymetrix, in looking at archival tissues and the effect of formalin fixation on the QuantiGene assay. So this is not tissue sections. This is showing HeLa cells that have either been fixed in formalin or not. And then we look at a gel, just looking at a northern gel, see that RNA is not degraded. When we look at control cells, cells that were not treated with formalin, 1 hour, and then 16 hours in formalin versus not. And the QuantiGene assay, this is our single-plex assay for 3 housekeepers that we're looking at here, actin, cyclophilin B, GAPDH. We know basically no effect of formalin fixation or the duration of that fixation. It's not the case with PCR-based assay, where we can see that CT values go up, meaning detected expression had gone down, that detected copy number had gone down when we compare 16-hour plus formalin versus no formalin, even 1 hour plus or minus formalin. You look at a more real-life situation, so clinical samples, in this case, they're looking at a fusion oncoprotein at the RNA level as well as the protein level. And sections were cut, serial sections, serial sections, one would be stained for the oncoprotein with immunohistochemistry. The other one would be H&E stain, and then some poor technician had to dissect out the area of tumor to separate it from the normal tissue, and then make the homogenate, as we would, and then compare levels of oncoprotein. So in this case, prostate cancer, there was huge concurrence in oncoprotein as well as the fusion gene for tumor expression. Another example of looking at QuantiGene as compared to different technologies for, again, formalin-fixed tissues. In this one, they were looking at large B-cell lymphomas, and their conclusion was that, they both work. The hands-on time for the QuantiGene Plex assay, again, is much less, and it simply worked better for these challenging type of materials. The panels that we have for the technology are very varied. You can go on our website and you can see a list of the type of panels that we've done for the different species, or you can just present us with your own list, and we'll make a QuantiGene Plex panel for you from that. People have been excited though about a new Immuno-Oncology QuantiGene Plex panel. A lot of hot targets on here. And, up till now, we've been talking about the Luminex assay for RNA. It works also for DNA. We're not even going to talk about that today, but sample preparation is a little bit different. We degrade the RNA, open up the double helix, and then we can look at 5 minutes left. Oh my gosh. Okay. We have information, I think, in the back about our ProCartiplex assays, which are the protein-based assays. So kind of working together with the QuantiGene at the RNA level, we can, with the Luminex platform, multiplex for proteins of interest, Immuno-Oncology panels, Immuno-Oncology checkpoint panels. I'm sorry, but I'm running out of time, so I just have to breeze through these. And then this was interesting because it combined the two assays. So we had cells in culture over time with stimulation. We sampled the supernatant, the media, and look at proteins, and we lysed the cells at the same time point to look at message. So here we're looking at protein IL-8, and this is mRNA for IL-8. You see maybe some levels of translational control. Each one of the different targets, cytokines that were tested showed different patterns. So it was very interesting, and it makes you think about, looking at your time points very carefully. We talked about QuantiGene single and multiplex. I'm going to see if in 1 minute I can go over view RNA.
Oh, you actually have 5 minutes to talk.
Okay, thank you. So we've been talking so far about, bulk measurements, averages across cells or tissues. But what about single cell analysis, and why would that be important to you? So, sometimes you need to know more than what the average is. You need to know if different cells are expressing differently within a population, different cell types, and also you might need spatial information within cells or within tissues about where expression is occurring. So, behind. For cell-based assays, we've done this for quite a while with a number of different products. Our newest one is called View RNA Cell Plus assay because it allows you to combine RNA with single molecule detection with protein with immunohistochemistry, so, or immunofluorescence.
Good to know.
So in this case, we're fixing and permeabilizing the cells. We're using our probe sets again to bind to targets specifically. We can look at 3 or 4 different RNA species in the same cells, looking at immunofluorescence for proteins. We're typically using about 1,000 bases that affords us single molecule sensitivity. So this is our demonstration of single molecule sensitivity. These are HeLa cells. We have probe sets for HER2. One probe set to the same message, but in different areas. One's going to light up in red, one's going to light up in green. We take an exposure with the red filters, move the microscope stage slightly, take a second exposure with the green filters, and we can see just about everywhere there's a green and red dot in the same orientation. So we're taking that as proof that we're seeing single molecules. If you've got no probes, daffy stain nucleus. If you've got a target that's not there, you see nothing. If you have a probe set to something that's there, you get dots that you can count and quantitate. If you do immunocytochemistry on its own as compared to Cell Plus, where we're looking also at RNA, the immunocytochemistry is the same, but we're getting the additional information from RNA. Older technologies use single labeled oligos to do ISH studies. We compared that to, well, we didn't do it, but to view RNA, and our signal amplification, our signal to noise, our low backgrounds are all far superior. We can do this in 384 well plates, time course. This is a quantitation of IL-8 dose response. Thermo Fisher has instrumentation for high content analysis, or it can be done microscopically. And we can work with tissues in a very similar method. The detection that we use is somewhat different. In this case, we are using alpha phosphatase and generating a precipitate, which in our first assay was red. So here we're looking at human insulin in obviously pancreas. The islet lights up in red. If you can't detect insulin in an ISH assay, you're in trouble. But the point here is that if you look at a no-probe control, you see 2 islets there, there's absolutely no signal. And here, there's no signal outside of where it should be. Radioactivity was the way to do it in the past. You never knew how long to expose. You had to make your own probes, and background was an issue, not what QuantiGene view it. We're going to have some nice data later. HER2 in human breast tumor versus normal tissue. And then, again, we can see as a single-plex assay, red dots here, not many in the normal tissue, and then expression levels confirmed with conventional QuantiGene. It works also on frozen sections. This is mouse brain and higher magnification. So we would say that each one of these dots represents a single molecule, becomes more semi-quantitative when expression is high. And we can do 2 colors now. So to summarize, QuantiGene assay is fast, simple, but most importantly, highly accurate. It's a lyse-and-go assay. It can do single-plex or multiplex. It can be applied to in situ hybridization for both cells and tissue, as well as for flow cytometry. Some of our customers, you guys should be up there too. And thank you so much.